Welcome to the Liver Meeting Debrief Session 2. Please welcome your moderators, Dr. Carla Brady and Dr. Debbie Shawcross.
Good morning, everyone. It's great to see you all on the fantastic session that we've got planned this morning. So we've got six really exciting debriefs. We're going to begin on basic science. We've got Yaron Rotman, who is a senior investigator and chief of the Liver and Energy Metabolism section in the liver disease branch of NIDDK, taking us through all of the best bits of basic science. I'm really looking forward to hearing what you have to say.
Good morning, everyone. Here are my disclosures. Essentially, I have no personal disclosures, but I did very well, twice. So when AASLD asked me to cover this, and I had to pick, I didn't know how difficult it's going to be to pick only a few abstracts out of the very many fantastic works that I've seen. I sort of felt like a kid in a toy store, or for those who know me better, a middle-aged man in a coffee machine store. And I promise this is going to be almost the last use of AI here. So to make this, just so you know what I'm going to do, I'm going to very, very briefly summarize some works. I'll tell you what's the question they asked. I'll tell you what is the model they used, because this is the basic science part. What are the very, very distilled key findings and why I picked it. And I'll always have the abstract number and the first author name on top. So if you find something interesting, please go and either talk to them, read the abstract, or send them an email. Okay, let's start. Portal hypertension. This is work presented yesterday by Dr. Ma from the Wakiri lab at Yale. And they the basis for this study is that we know that in portal hypertension, the decreased blood flow through the portal system is accompanied by a compensatory increase in the hepatic artery flow. And they asked a very smart question, does that matter? Does that affect liver remodeling? The model they used is a rat with partial portal vein ligation, so they essentially can control the diameter of the portal vein and control the degree of portal hypertension. So what did they find? First they found that the model worked, so they did see the compensatory increase in hepatic artery flow, and they found that there was upregulation of VCAM1 on the endothelial cells in the hepatic artery. Remember, they did not do anything to the hepatic artery. This is just because of the flow. They then could see that this leads to recruitment of specifically osteopontin-secreting macrophages, which they could also demonstrate the presence of in human samples with portal vein thrombosis, and that we know that osteopontin is a major activator of stellate cells and leading to fibrosis. And finally, when they treated their animals with anti-VCAM1, they could actually decrease the degree of fibrosis that is seen. So I thought that was interesting because we found that in portal hypertension, the increased flow in the hepatic artery is sufficient by itself to drive portal fibrosis through the VCAM1 macrophage axis, and maybe VCAM1 inhibition could be a therapeutic target in some of those people. So moving from pre-hepatic to post-hepatic portal hypertension, this is a study by Dr. Kato from the Takehara group at Osaka, and this was looking into hepatic congestion and how does that lead to fibrosis. They used the mouse model and partial inferior vena cava ligation. And what they found in the model is that after the ligation, specifically in the zone 3 liver sinusoidal endothelial cells, they see upregulation of integrin alpha V, leading to activation of the YAP-TAZ pathway, secretion of CTGF, and that CTGF can activate the stellate cells and drive fibrosis. Then they very nicely were able to show that if they knock out CTGF, specifically in the endothelial cells, they can improve the fibrosis, they can improve the portal pressure, and even prevent some of the cancers that develop in these animals. And they achieved the same results by treating them with an anti-integrin. And they did have a correlate in human samples from Fontan-associated liver disease. So I thought that this was a very nice way. They've shown how the pressure itself, hydrostatic pressure, leads to changes in the endothelial cells that eventually drive a fibrotic process. And maybe we can use anti-integrins, for example, in Budd-Chiari syndrome or something like that to prevent damage or decrease damage. Let's move to cancer. This is a study shown yesterday by Dr. Lemaitre from the Danextekaran group at Stanford. And so we treat some patients with hepatocellular carcinoma with transarterial chemoembolization. We know that in most cases this is not curative and the cancer recurs. And the question they ask is, how come those residual cells that then eventually become a recurrence are not hit by the immune system? What helps them escape the immune detection? So they started their model with generating doxorubicin-resistant hepatocellular cells and growing them in 3D co-culture organoids with macrophages. And they also used transplant of human cancers into mice. What they found in their organoids is that the resistant hepatocellular cancers change the phenotype of the macrophages to a more pro-tumor phenotype with expression of PD-L1 and secretion of TGF-beta. And then they used their mouse models, so they took mice, they transplanted them with doxorubicin-resistant HCC, and they treated them hitting both pathways with TGF-beta inhibitor and anti-PD-L1, and there was a remarkable reduction in the tumor burden in these animals. So this is a nice pathway to suggest how residual HCC aftertaste maintains immune evasion, and potentially suggesting an adjuvant therapy in humans in the future. This is a work by Dr. Zhang from Seoul National University, and we will talk about DPP4. Most of us, or the clinicians, know DPP4 from the diabetes field. So DPP4 is a peptidase that cleaves proteins and basically regulates protein levels by cleaving them. So something I didn't know until I read that abstract, that DPP4 also cleaves CXCL10 and inactivates it. And CXCL10 is a major chemokine that with roles in both infection and anti-tumor activity. So then they ask if this is DPP4 can impair CXCL10, maybe hitting DPP4 can help us in a cancer model. So they used mice. The mice were injected with HCC, and they were treated with either anti-PD-1 regorafenib, or sitagliptin, which is a DPP4 inhibitor, or the combination of both. And very simply, you see a marked effect. So black is the control, where red is the combination therapy, and this is the tumor burden over time, and this is the tumor weight. So there's a remarkable effect when you, as an add-on inhibition of DPP4. And they also saw, I missed that. So they also saw an increase, which you'd expect, the increase in effector immune cells into the tumor. So this suggests that DPP4 inhibitors may enhance the response to immune checkpoint inhibitors in patients with HCC. And DPP4 inhibitors are clinically available, are safe. I think they're going generic, so maybe this is applicable to humans. Okay, alcohol-associated liver disease. This is a study that was brought to us by Dr. Nagesh from the Zhongyi Sabos group, and from Beth Israel. And I completely forgot to mention, if you see this icon, this is work that was supported by the AASLD Foundation, so please donate. We know that alcohol-associated hepatitis has several important features. There's steatosis, there's a lot of inflammation related to neutrophils, there's an activity of the NLRP3 inflammasome. This is a lot of work that's been shown by that lab over the years. And the question is, can we have one target to rule them all? Can we hit all of those with one thing? And the authors suggest that Bruton's tyrosine kinase may be the candidate, and to do that, they've used several models. They've used samples from human patients with alcohol-associated hepatitis. They used the mouse model, the classic NIAAA of chronic exposure with binge, and they used the evobrutinib, which is a BTK inhibitor. So first, they could see that in human and mouse samples from alcoholic hepatitis, they see upregulation of BTK phosphorylation. And in vitro, they can see that this is directly affected by ethanol, that ethanol induces the phosphorylation, and that the phosphorylated BTK will stabilize the NLRP3 inflammasome. They then do the mouse experiment. They treat the mice with BTK inhibitor, and you can see that there's a marked reduction in damage as suggested by ALT, in triglyceride accumulation, as well as they could show that there's a decrease in macrophage NLRP3 and a decrease in neutrophil netosis. So overall, it does seem that the BTK inhibitor can improve multiple separate features of alcohol-associated hepatitis in a mouse model. And again, these inhibitors are available clinically. They're currently used in cancer treatment, so maybe they will work in humans. I think it's at least worth trying. This is another study on alcohol-associated liver disease coming from a collaboration between Elena Kaff at Penn and Waj Mahal and Richard Flavell at Yale. And they were asking about the role of lipids in alcohol-associated hepatitis. So what they did is they took blood samples from humans with alcohol-associated hepatitis, heavy drinkers who don't have hepatitis, and healthy controls. And what they did lipidomics on those blood samples, and what you can see very broadly, without even looking at details, is the patterns are very different. There are several lipid species that are markedly upregulated and specifically in alcohol-associated hepatitis. Then they did something very cool. They used a humanized mouse model, but the problem with most humanized mouse models is that they don't have a good, they're immune deficient. And this is actually an immune-competent humanized mouse model because they transplanted the mice with both human hepatocytes as well as with human cord blood, so you actually get the human immune system in them, at least partially. They exposed these mice to the NIAAA mouse model, and they actually see the same alteration of lipids that they found in the humans. So this makes it a useful model. Then they asked, are these lipids just a feature or are they causative? And what they did, they used regular mice, not humanized mice. They exposed them to the alcohol model, and they also treated them with those specific lipids that they saw upregulated in the humans. And what they found is that they could recapture some, not all, but some of the features of worsening disease. They had a higher ALT in some of those lipids. They had the similar immunological features in the livers. So I picked this because I thought this is a very cool mouse model and it seems like a useful model to study that disease. And also because it's interesting that it suggests that the lipids that we see altered may actually have a causative role in promoting the disease progression. Okay, let's talk about AASLD. So this is work from Dr. Daniel from the Harmeet Malhi's group at Mayo Clinic. And we know that if we look at the hepatocyte in MASH, the hepatocytes are miserable with lipotoxic ER stress. There's an entire epigenetic regulation and change of hepatocellular processes probably to accommodate for that. And also that those miserable hepatocytes are releasing damage-associated molecular patterns through extracellular vesicles that affect other cell types and affect the rest of the liver. And a lot of this comes from Harmeet's previous work. And how are all of these connected? So what they did is they used three models. They used extracellular vesicles from in vitro hepatocytes cultured with palmitate to generate lipotoxicity. They took livers from the classic mouse models of MASH or MASH fibrosis, and they also had patient sera. And in all of those samples, they could see specifically upregulation of a protein S100A11. Here I'm just showing you extracellular vesicles from the in vitro experiments and how it's upregulated by palmitic acid. Now comes the clever part. So they went into the genomic region upstream of near S100A11, and they found a putative enhancer site. And they could show that that enhancer site is specifically acetylated when exposed to lipotoxic stress. So they promptly named it the lipotoxicity influence enhancer, or LIE. Then they had a very nice mouse model based on CRISPR-Cas technology, where you very, very specifically block the ability to acetylate that enhancer. It's the Cas9 crab mouse model, for those who want to read about this. And when they block just that specific enhancer acetylation in those mesh models, they can actually see that there's a decrease, as expected, in S100A11 secretion, but also improved ALT and improved fibrosis. So this tells us several things. First, they actually have a very nice pathway how lipotoxic stress and through something I haven't shown here, through the ER stress, cleavage of and activation of XBP1 that migrates to the nucleus, very specifically activates a very specific site, I said specific too many times, enhancer for S100A11, and they also show that S100A11 may actually play a role in the pathogenesis of the disease. Okay, let's talk about toxic injury, or more specifically, acetaminophen. So this is coming from Dr. Kay from the Wenshie's group at University of Pittsburgh. And we know that in the liver, after acetaminophen injury, there's marked oxidative stress and injury. Now, proteins have different responses to oxidative stress, and one of the processes is S-sulfinylation, the attachment of a sulfinyl acid to proteins in the context of oxidative stress. And this is a transient thing. It gets attached and then it gets removed. The major desulfinylation enzyme is sulfirioxin-1, SRXN1. What is the role of the process and what is the role of SRXN1 in acetaminophen injury? So the authors first look at samples from both humans and mice treated with acetaminophen, and they found upregulation of S-sulfinylation, and they found, as in this immunohistochemistry, upregulation of SRXN1 in those livers. They then generated a SRXN1 knockout mouse, and what hepatocyte-specific knockout mouse, and what they could show is that if you knock out SRXN1 and expose the mice to acetaminophen, you actually have way worse injury. And the flip side is that if you overexpress SRXN1, you actually improve the injury. So what this is suggesting is that the sulfinylation of proteins in the context of acetaminophen-induced oxidative stress is damaging, that the upregulation of SRXN1 is a compensatory pathway to actually try and control that and contain that damage, and that maybe if we target SRXN1 some way, we may be able to help these patients. Another study looking at acetaminophen injury was brought to us by Dr. Jain from the Bushan lab in University of Pittsburgh. And they were focusing on the EGF receptor on hepatocytes. So we know that the EGF signaling and EGF receptor are crucial for hepatocyte proliferation, and especially in models like partial hepatectomy, where you need the liver to regenerate. Interestingly, in previous studies, the regeneration that happens after acetaminophen injury, you also need the hepatocytes to regenerate, seems to be actually inhibited by the EGF signaling. So they wanted to look at and explain this, and they used an hepatocyte-specific EGFR knockout mice, and they treated them with acetaminophen. And what they found is that their knockouts actually had remarkable improvement in resolution of injury. You can see that the wild types, this is the ALT and the wild types over time, and you can see that in the knockouts, the injury actually peaks at 12 or 24 hours and then decreases. And this is accompanied by remarkable recovery of glutathione stores, and we know that, so basically they just recover better. Now what's the mechanism? They very nicely show that in the knockout animals, there's a decrease in phosphorylation of Jun kinase and its translocation to the mitochondria. And that leads first to less mitochondrial damage, but also there's upregulation of mitophagy. So the damaged mitochondria can be handled in a safe manner. And that drives increased and faster regeneration. And of course, then they treat the animals with EGFR inhibitors, and they can show that these animals have benefit both if you treat them very, very early, or even if you start treatment 4 hours after the exposure, which is a little bit more like the clinical feature that we tend to see. So overall, I thought it's interesting because they show a pathway that EGFR, although it's useful in regeneration after hepatectomy, actually contributes to the damage, and they show how by preventing the containment of mitochondrial injury. And maybe we can use EGFR inhibitors in the future in people who have acetaminophen overdose. Okay, let's go to the biliary system. This was very nice work coming from Dr. Ferreira from the Forbes lab at the University of Edinburgh. So we heard a lot about senescence in the basic science presentations in this meeting, and it's been known that in biliary diseases, you can see a senescent phenotype in biliary epithelial cells. And the authors ask a very interesting question: does that actually affect the hepatocytes as well? So first they looked at human samples, and they looked at human samples from biliary diseases. We have biliary atresia, we have PSC, we have PBC. In all of those, they saw senescent biliary epithelial cells that express P21. But also in all of those, they saw that hepatocytes upregulated and expressed VCAM1. We've already spoken about VCAM1 earlier. And you can see it here in purple around the hepatocytes in all of these three models. And they also found that there's clustering, that there's migration of neutrophils and clustering specifically around the senescent biliary epithelial cells and specifically around the VCAM1 hepatocytes. They're here in yellow, the neutrophils. So then how does that work? They go in vitro, they generate senescent biliary epithelial cells, they co-culture them in a non-contact system with primary hepatocytes, and they see that this is sufficient to induce the senescent phenotype is sufficient to induce VCAM1 expression in the hepatocytes and also to induce and to upregulate programs that are associated with neutrophil recruitment, and they could then replicate it in a mouse model. So overall, I think they show a nice pathway where the senescent biliary epithelial cells signal to adjacent hepatocytes upregulate a VCAM1, which then sort of calls the neutrophils to the region, and the neutrophils actually can target those senescent biliary epithelial cells. This could be beneficial or could actually be a harmful feedback loop, and maybe if we can target this biliary hepatocyte communication in some of the biliary diseases. From chronic to more acute, this is data by Dr. Kutsenko from Paul Monga's lab. So we know quite a lot about the role of beta-catenin, especially in the context of Wnt signaling. And and maintaining hepatocyte identity, and a lot of it actually comes from the Munger lab. But beta-catenin also has another role. Together with its cousin gamma-catenin, they actually form the adherence junction on biliary, in biliary epithelium, keeping those cells together. And the question is, how important are they there, and what's the importance of the adherence junction? So the authors used a double knockout mouse model where they knock out specifically in biliary cells, in biliary epithelial cells, the beta and gamma-catenin. You have to do this in an inducible system because otherwise they would never develop. So this is inducible. The knockout is driven by exposure to tamoxifen, so you can actually control it. So you take these mice, you give them tamoxifen, you knock out beta and gamma-catenin, and then in 4 weeks all of them die. And when you look in their livers, you see tremendous biliary infarcts. There's complete loss of integrity of the biliary system. This is dye injected into the biliary tree, and you can see how it leaks in the damaged animals. They also could show it functionally, that there was a change in an altered biliary flow in these animals. But all of these animals die. You want to study how to recover, so they could use the benefit of the tamoxifen-induced system to actually titrate the damage. So they used a milder version. They did not completely knock it out. And then some of the animals died, but some of the animals actually are able to recover. And when you look at those animals, what's very, very interesting is that you actually see changes in the hepatocytes. And some of the hepatocytes are starting to express biliary markers like A6, which is shown here in the figure. So although this is preliminary, this is, I think, very nicely shows the roles of the beta and gamma-catenin in maintaining bile duct integrity. It gives us a useful model for acute biliary injury that can be titrated. And it shows the importance of the transdifferentiation in hepatocytes and their plasticity in helping to recover from something that's actually starting at the biliary tree. Okay, viral hepatitis. So this is a study by Chloe Berm, a medical student from Charlie Rice's lab. And very simply, they wanted to identify host factors that regulate viral HBs antigen production and maybe could be used as targets in the future. They were specifically focusing on stages that are after the viral entry into the cell. And this was done by transfecting HUH7.5 hepatocyte cells with HBV pregenomic RNA. So this is you transfect at this step, you bypass all the entry sites, and you just go, you only study these levels. And then they use those transfected cells in essentially in an automated manner with a CRISPR-Cas library that individually knocks out every single gene in the genome. And you want to see what every single gene in the genome, what that would do to HBs antigen expression. So this is a very broad screening method. And looking at all of the genome, they found some targets that are increasing the secretions, and these are, when you knock them down, so these are antiviral, some targets that are decreasing secretions, these would be porviral. Then they they use a confirmatory assay of the 100 top hits, and they could confirm many of them. They found proviral, antiviral, they even have very nicely NTCP, which is the entry, and you expect it not to be changed because that's not the step that they use, and they could confirm this. And unfortunately, we don't have more data on the specific targets, but I thought it was just a nice start to something that may lead to new therapeutic targets. Okay, and we're going then to regeneration. So this is work that was presented by Professor Kaur from the Institute of Liver and Biliary Sciences in New Delhi. So we know that when you regenerate in the classic model, the liver regenerates, a classic model is after partial hepatectomy. It's a multicellular event. The hepatocytes regenerate, but you need to build new blood cells, you need to have all the non-parenchymal cells. We actually don't know much about the liver lymphatics in that context. So the authors used a model of rats, undergo 70% partial hepatectomy, or a sham experiment as a control. And then they specifically looked and stained the lymphatic endothelial cells in these recovering animals, and you can see them in red. And what they found is that the kinetics of recovery really paralleled the kinetics of recovery of cholangiocytes. Here you can see them in green, of the biliary tree. And when they did single-cell proteomics on these recovery lymphatic endothelial cells, they could see that there's upregulation of Wnt7a. We spoke about Wnt signaling just recently. So Wnt is a secreted factor so do they secrete anything? So then they take lymphatic endothelial cells, They grow them in culture, they use conditioned media, and they treat cholangiocyte organoids with that conditioned media, and they could show that that actually increases the proliferation of these organoids. And then they go back to the animal experiment. They block lymphangiogenesis after partial hepatectomy with anti-VEGF treatment, and they saw that this truly decreases the recovery of cholangiocytes and biliary tree. So overall, I thought that at first, this is a good study to show us that we tend to neglect the lymphatics in the liver, but this is how important they are and how they recover after partial hepatectomy. But also that this is not just the lymphatics recovering, that again, we have this cross-cell type communication that's important for other things. And for the final abstract of the morning from me, this is work coming from pharma, presented by Dr. Lee from Regeneron. And this is dealing with humanized mice. So for those of you that don't work in humanized mice, these are essentially mice that are A, immune deficient, and B, there's something in them that kills the hepatocytes. People have used in the past TPA, and now I think most people are using the FAH knockout, which essentially is a model of tyrosinemia, and you can keep them alive until you change their diet, and then their hepatocytes die. And what people do is then you inject them with primary human hepatocytes that will then go home into the liver and engraft. So you get a mouse with human hepatocytes. That sounds fantastic, but these are very difficult models. The engraftment can be slow, can be unpredictable, you get a lot of variability, and you have some donors that are not very good donors, you see that they don't engraft well, and also the engrafted hepatocytes tend to develop spontaneously lipid droplet accumulation, which suggests that they're not very happy. Now, they had a very nice hypothesis, and they said, okay, you know, we have those human hepatocytes in the mouse, but the signals that they see are mouse signals, they're not human signals. So maybe we have an issue with mitogenic signals. And they show it very nicely here, where you take human hepatocytes, and if you treat them with human IL-6, you see that the pathway that, for example, this is STAT3 phosphorylation, is affected. But if you treat them with mouse IL-6 or rat IL-6, you don't see that. So essentially, we put the hepatocyte there, but it doesn't see the signals it's used to. So can they improve on the process? So of course they can. What they did is they took this model, which is like triple or quadruple knockout, and they genetically modified them even further. So they made these mice express the human IL-6 instead of the mouse IL-6. And then hitting another mitogenic pathway, they treated these animals with c-Met activating antibody, a human c-Met activating antibody, which Regeneron has. And when you hit both of these targets together, not each one separately, this is what you see. You can see that when you hit both targets together, you have a marked increase in human albumin in the serum. And if you look, the cells stained in brown here are stained for human albumin. And you can see that in this liver, there are a few human hepatocytes, but here there are way more. Then they even go even further. Instead of hitting 2 pathways, they hit 3, and they treated these animals with human endothelial growth factor. We just recently spoke about that. And you can see really, this may be a typical best example, but still you can see marked increase in human albumin and engraftment. So I thought this is interesting because without repeating the word humanized too many times, this is actually a way to improve the model, make it more effective. And these are models that are very helpful in studying human disease in a mouse model. I'll end first by thanking all the authors for fantastic studies, not only the ones that I presented, but also other studies, and for sharing their slides with me. AASLD staff for help, AASLD Foundation for paying for a lot of this work. I also would like to apologize to the authors whose work I butchered and the authors whose work I couldn't show, to the audience who had to tolerate me. And I'll return to AI and say that if you could handle this until now, then you're welcome to enjoy your coffee. And it's quite remarkable what you can do with those software. So thank you, everyone.
Thank you very much. Next, I'd like to invite to the podium Dr. Tatiana Kushner, who is an associate professor of medicine in the Division of Gastroenterology and Hepatology at Weill Cornell School of Medicine. She will present this year's hepatitis debrief.
Well, thank you so much to the organizers and to AASLD really for the honor of presenting this year's viral hepatitis debrief. It has been a very exciting time in viral hepatitis. This year we had approval of point-of-care testing for hepatitis C. We have now recommendations in the US for universal hepatitis B screening. And at AASLD this year, we saw many very engaging sessions on elimination, on our efforts towards getting there, and of course on therapeutics. This is a brief outline of what I'll cover, but mainly I'll focus on hepatitis B, Delta, and of course hepatitis C. So I'll start with just one abstract that I'll highlight that addresses hepatitis B epidemiology and natural history. I thought this was an abstract that's relevant to our clinical practice and our current guidance on hepatitis B treatment. So currently, the recommendation is to treat individuals with hepatitis B viral load over 2,000, and we don't routinely treat those who have hepatitis B viral load under 2,000. And this abstract addressed two distinct groups in that population, those individuals who have low-level viremia, meaning viremia that's between 20 and 2,000 IUs per ml, compared to those with strictly undetectable hepatitis B DNA, which is less than 20. This was a multi-center retrospective study comparing significant clinical outcomes in these two patient populations, which included progression to e-antigen negative indeterminate phase, which means HBV DNA over 2,000, surface antigen clearance, as well as progression to significant liver fibrosis and cirrhosis. And what they saw in these two distinct groups is that there were distinct clinical outcomes as they followed them over time. They saw that there was a higher likelihood of hepatitis B surface antigen seroclearance in those who had undetectable HBV DNA, and a higher likelihood of progression to the indeterminate phase in those with low-level viremia. So this suggests that perhaps our treatment goals should be more aggressive and that we should really be aiming for undetected HBV DNA levels and really in prognostication know that these are distinct groups that may have different outcomes. So now I'll shift gears to hepatitis B and the interesting studies that were presented this year using approved therapeutics. This is a study that looked at a very important patient population. This is the pediatric patient population with hepatitis B. And really the guidance about treatment in pediatrics is not entirely clear and varies. And this was a study that sought to investigate the efficacy of safety of antiviral strategies in the pediatric patient population and to explore factors associated with hepatitis B surface antigen loss. This was a prospective multi-center real-world study done in China in children aged 3 to 18, addressing two strategies for treatment that were selected by the families and which chose which strategy they hoped to pursue. One of them was pegylated interferon alpha 2B combined with NA monotherapy, and the other one was NA monotherapy alone. And in the study, they looked at almost 300 children and followed them over time. The goal for the study is treatment for 96 weeks, and here they reported outcomes at 48 weeks of treatment. And what you see, not surprisingly, is that in the combo arm, the kids did better. There was a 25% likelihood of surface antigen loss in children that were treated with pegylated interferon combination. Furthermore, when they stratified by age, it's the younger children that actually had higher rates of surface antigen loss, so 31% versus 20%, and those children that were under age 7. So the conclusion here is that hepatitis B infected children can achieve significantly higher surface antigen, e-antigen loss in treatment with pegylated interferon. Of course, the first question that came up in my mind is how do they tolerate this treatment? And actually, it appears that children tolerate pegylated interferon better than adults, and they really tolerated the treatment well, even for very long durations. Another study now shifting to adults also addressed the use of pegylated interferon in combination with tenofovir in an adult patient population. And these were results from a multi-center randomized controlled phase 3 clinical trial, also conducted in Asia. And here they had 4 arms for treatment, 3 with NA experienced patients and 1 with treatment naive patients, and compared tenofovir alone compared to different arms of pegylated interferon at the 180 and the 90 milligram dose. And what they saw here is that in almost 400 individuals in the full analysis set, that again, those individuals that were treated with pegylated interferon had significantly higher, significant rates of surface antigen loss at around 30%. What they also saw is that baseline surface antigen titers are very important in predicting in who will have surface antigen loss. And so those with surface antigen titers of less than 100 were more likely to achieve surface antigen loss, which we'll see as a recurring theme as we move to novel therapeutics. Notably, I didn't mention, but this study looked at 24 weeks post-treatment. So these are results of surface antigen loss after 24 weeks being off any treatment. So their conclusion here was that pegylated interferon alpha combined with tenofovir can achieve durable surface antigen loss. Again, the question of tolerability comes up. Here, they used actually an intermittent treatment protocol where they took treatment breaks throughout the duration, which helped with improving tolerability for the patients that were on the pegylated interferon. And the last abstract I'll present in regards to approved therapeutics is the treatment of the pregnancy patient population. So this evaluated tenofovir alafenamide in pregnant individuals at a shorter duration of 8 weeks compared to a longer duration of 12 weeks. Of course, in the pregnancy context, we like to minimize medication exposure if at all possible. So here they looked to see if in those pregnant women with high level viremia, if we can use 8 weeks of tenofovir alafenamide and achieve the same results. The main endpoints here were safety as well as mother-to-child transmission. And what they saw here in the per protocol analysis is that there was no mother-to-child transmission in either of the groups. In the intention to treat, you see some reported mother-to-child transmission, but that was really due to loss to follow-up as opposed to actual events of transmission. And so the conclusion here is that maternal TAF prophylaxis to prevent mother-to-child transmission is generally safe and effective, and we can use shorter duration of 8 weeks, which is feasible. Recently, this week, also Calvin Pan published a paper in JAMA that actually looked at longer duration antiviral therapy, but without the HBIG immunoprophylaxis. So I think as we move forward, we'll have individualized treatment regimens in the pregnancy population depending on region and accessibility for antiviral therapy and immunoglobulin. Now I'll have a few slides about hepatitis B novel therapeutics. This is a very exciting space, and this is a very busy slide that probably is not even entirely up to date, but just to show how much drug development is going on in the hepatitis B and Delta space. And you see here that we have phase 1, phase 2, and phase 3 drugs that are in development, and that really we are looking at them as two categories of medications: the direct-acting antivirals, where we're seeing a lot of siRNAs, capsid inhibitors, and then the immune-targeted therapies, such as monoclonal antibodies, checkpoint inhibitors. In phase 3 of drug development, we have 2 medications currently, which is very exciting. This is the first time in a while that we have phase 3 drugs available, and we have bepirovirsen, or Bepi, for the treatment of hepatitis B, and we have bulevirtide for the treatment of Delta hepatitis. And as many of you know, bulevirtide is already approved for use in Europe and is actively being used for the treatment of Delta hepatitis. So just a few studies about the novel therapeutics that updated data was presented at this meeting. The first one is from the company Vir, and it looked at a combination regimen with tobevibart, which is a monoclonal antibody, and elebsiran, which is an siRNA. So again, we're looking at combining a direct-acting antiviral and immune therapy that have complementary antiviral and immunomodulatory effects to hope to achieve functional cure. The goal here was to investigate the safety and efficacy of this combination regimen with or without pegylated interferon. So we are seeing that pegylated interferon is still playing a role even in these novel therapeutic regimens. And what they did is they had 3 arms which looked at monotherapy, dual therapy with these 2 agents, and then combination with interferon. And the results presented here are end of treatment. So these are not off treatment results, these are end of treatment results. And what they found is that in the individuals on the combination therapy, there is a 16% likelihood of surface antigen loss at end of treatment, and a higher rate at 22% in combination with pegylated interferon. So the conclusion here is that these agents in combination with pegylated interferon, possibly without, are promising, and follow-up is ongoing to determine functional cure rates after cessation of study treatment, which will be really important to see. The next study that I'll share updates on is from Arbutus. This is also a combination therapy with an siRNA, imdusiran, which was used every 8 weeks with nucleoside analog therapy, leading up to a combination with a 24-week course of interferon. We already saw earlier in the year results at end of treatment, but here they presented treatment results at 24 weeks after end of treatment. They had 43 non-cirrhotic, NA-suppressed, e-antigen negative subjects that received initially 4 doses of this imdusiran and then potentially received 2 additional doses for a total of 6 doses with interferon, followed by a consolidation period of nucleoside analog therapy alone. And again, these are results off treatment 24 weeks after treatment completion. And what they found, which is very exciting, was that in the group that received the 6 doses of the siRNA imdusiran, 25% of the individuals achieved surface antigen loss. And in those that started with surface antigen less than 1,000, 50% achieved functional cure. On the right, you see the viral kinetics pictured, which show that most of the individuals that had surface antigen loss also mounted surface antibody. So very encouraging data, and it will be important to see how they do further out after treatment. And I'll just mention one quick abstract that I just saw recently that was presented yesterday, and this is really investigating a novel immunotherapy. So many of the data and abstracts that I shared until now included interferon as part of their treatment regimen, and really a question in the field is, will we stay with interferon, or will there be other effective immunotherapies that can take the place potentially of interferon? So VTP-300 is a targeted investigational immunotherapy designed to elicit AG-specific T cells. And this was a phase 1b/2a study that showed sustained reductions in surface antigen when given alone. And in this particular study, they combined it with a low-dose checkpoint inhibitor. They did see some promising early data that there was surface antigen loss in some of the individuals on these treatments. And the conclusion here was that this preliminary safety data showed that VTP-300 in combination with low-dose nivolumab is well tolerated. There were no significant treatment-related adverse events observed. And perhaps this may be an important component of future functional cure regimens. So now let's shift to delta hepatitis. A lot of abstracts this year on delta hepatitis. We saw many updated looks at local, regional, national, global epidemiology. I'll share just a few of them here. There was one abstract that was from the country Georgia, where they looked at prevalence of delta among hepatitis B positive individuals in Georgia, and this was a national seroprevalence survey. They found a 3.7% HDV positivity rate among samples that they tested from this national survey. Similarly, there was a national study from Canada that looked at prevalence from referred versus non-referred specimen that were sent to their national lab, and saw 4.2 versus 1% HDV positivity. And then there was updated data from US database from the US veterans, which looked at prevalence, but also association with long-term outcomes. This was a large retrospective study which identified a 3% prevalence of delta among patients with hepatitis B, very high prevalence of substance use among these patients with delta hepatitis in this US-based cohort. And they saw a significant association with long-term liver-related events, including HCC, hepatic compensation, all-cause mortality, and liver-related mortality. And really, one of the key messages that we've seen here in the US is that we're really not testing enough in order to be able to link these patients to care and optimize their outcomes. We presented data from our system in terms of implementing reflex testing, which is one potential solution. There were significant barriers in getting this done, but it was effective in improving our delta hepatitis testing rates within our health system. So now let's turn to therapeutics for delta hepatitis. So we saw many abstracts at this conference on bulevirtide, probably maybe 10 abstracts on bulevirtide, both clinical trial outcome data as well as real-world data that we're now accumulating largely from Europe. This is the SAVE-D study. This is long-term bulevirtide monotherapy in patients with HDV-related compensated cirrhosis. So this is a group of individuals with cirrhosis that many of them had a history of ascites, varices, even a few with active HCC. And this is long-term, long-duration treatment up to 120 weeks in patients with delta hepatitis and cirrhosis. And they saw really positive results with 120 weeks, 74% having virologic response and almost half having combined response virologic with ALT. Furthermore, given the long-term follow-up, they were actually able to look at clinically relevant endpoints, including HCC and hepatic decompensation. They saw low rates of these events, and another poster compared events in the bulevirtide-treated arm compared to those who were not treated, and found that there was a trend towards decreased hepatic decompensation in individuals on bulevirtide. So this is very promising to see that long-term duration therapy with bulevirtide is well tolerated, even in patients with cirrhosis. In regards to other novel therapeutics, there was updates from the SOLSTICE trial. So this is week 24 primary endpoint analysis from the VIR combination, which is, again, topivirtide, monoclonal antibody, and an siRNA. Here they randomize individuals one-to-one to either mono or combo therapy versus rollover, meaning they started with monotherapy, then rolled over into combination therapy and had longer duration follow-up. And here they present week 24 results of the two top arms and then week 60 results of the rollover arm. Again, we're seeing very promising results in terms of endpoints. So 100% of individuals had HDV RNA response. And then in regards to the combined endpoint, high rates that were sustained even in the combination rollover arm that showed that longer duration of treatment has sustained response. So very exciting that this combination regimen is now planning to move into phase 3 with the phase 3 trial to start in 2025 for the treatment of delta hepatitis. The last agent that I'll present on is the Blue Jay agent. So this is a monotherapy for delta hepatitis, a monoclonal antibody, BGT778. The goal here, again, was to evaluate safety, virologic response, and ALT normalization and combined response in patients with HDV. They had 3 courses of treatment with this monoclonal antibody. And what you see, again, is promising results that over time, there was increase in virologic response, including to target not detected in all 3 arms, as well as promising data regarding up to 75% combined response in these individuals. All right, so in the last 3 minutes, I'll move to hepatitis C. So just an update here about how we're doing in terms of our efforts towards hepatitis C elimination. This was an abstract presented by Tashali from the CDC, and it looked at our treatment rates to date with direct-acting antiviral agents using the IQVIA database. So overall, the estimated number of individuals that have been treated in the US with DAAs is estimated to be about 1.3 million. Treatment rates peaked in 2015, soon after the DAAs became available. And although we're really not close to achieving our hepatitis elimination goals, we are seeing some promising trends where there are an increased number of individuals that are being treated that are covered by Medicaid. In addition, although most individuals who are treated are over age 60, we're seeing a rise in treatment of younger individuals. So the conclusion here was that despite overall low treatment numbers, increase in the number of younger persons and those covered by Medicaid is encouraging. However, we really need to make more efforts to get to our elimination goals, which includes actual implementation of the newly approved point-of-care RNA testing, removal of any Medicaid restrictions and specialist provider restrictions, and innovative programs to reach young individuals with hepatitis C. So not too much in regards to novel therapeutics, but a lot that we saw in regards to novel therapeutic settings and important new therapeutic settings for the treatment of hepatitis C. So this was a study presented by Faladi Nwulia and colleagues, and this was a multi-center randomized controlled trial that evaluated the difference between treatment on-site in an OTP program and standard of care off-site. And what they saw is that there was a clear benefit in hepatitis C rapid treatment initiation in those that were treated on-site compared to those with standard of care, as well as higher SVR rates. So this is the type of strategy that really we should try to think about moving forward in order to optimize treatment and working towards elimination. In addition, there was another abstract by Tony Martinez and colleagues that looked at same-day rapid start of hepatitis C treatment, which also was very encouraging in terms of our ability to achieve SVR in difficult-to-reach populations. The last abstract that I'll present is abstract about treatment in what I think is the last frontier patient population, which is the pregnancy patient population. This was interim results from the STORK study, which is a multi-center phase 4 open label study for hepatitis C treatment in pregnant individuals from 20 to 30 weeks gestation. And this data was very promising. We're halfway done with our recruitment goal of 100 individuals with 50 patients that are currently enrolled. Of the patients that completed follow-up, 100% achieved SVR12. There were no significant adverse events related to the drug, and notably there were some benefits. For example, 0% of individuals had intrahepatic cholestasis of pregnancy, which is an associated issue with hepatitis C, suggesting that there's actual clinical benefit aside from hepatitis C cure in these individuals. One question that came up is who will take on treatment of pregnant individuals moving forward? Will it stay in the obstetric realm, or will hepatologists do this? And so we have an ongoing survey that all of you are welcome to participate in to share with us if you would be willing, given the expanding data, to treat hepatitis C in pregnancy. If you are treating hepatitis C in pregnancy, there's also an online registry that you can submit your cases to as we continue to accumulate real-world data. Just a few additional late breaker abstracts that I wanted to quickly highlight, that there are other agents in the hepatitis B space. Unfortunately, I couldn't cover everything. Another agent called Viron, which is an immune therapy, as well as capsid assembly modulators are actively being investigated as another mechanism. With hepatitis C, we saw other abstracts, one focused on the use of a glecaprevir pibrentasvir, multi-center single-arm prospective study in the acute hepatitis C patient population, as well as another abstract that looked at difficult-to-treat patient population with genotype 3 compensated cirrhosis. So I'd really like to thank all of the presenters who made the presentations available for inclusions. Really like to thank the people listed here for helping me bring together this debrief, and thank you very much.
Thank you very much, Dr. Kushner. So we're going to move on to hepatocellular carcinoma now, and I'd like to invite Dr. Nihar Parikh to the stage. He's an associate professor of transplant hepatology and medical director of the liver tumor program and director of hepatology at the University of Michigan. So over to you.
Thank you very much, and I want to thank the organizers for allowing me to present today. My voice is a little off, so I apologize for that, but it's a cold. It's not what you think it is. So again, I'm from Nihar Parikh. I'm from the University of Michigan. I'm pleased to really present the HCC debrief. It's been exciting advancements in the field. So I categorize this in different ways here, and you can see the abstracts that are included, including risk prediction, surveillance, prognosis, and HCC treatment, and we'll kind of go into depth on all of these different abstracts. So to start, we'll start with risk prediction for HCC, and the first abstract is from Vujkovic et al. From the University of Pennsylvania. I just want to remind everyone that risk prediction and risk stratification has several benefits, and it's a hot field right now in HCC in terms of surveillance, because you can provide high-intensity surveillance for higher-risk patients, and there are additional benefits to minimize surveillance-related harms. You can do things like risk modification in high-risk groups, and these patients are primed for chemoprevention if you can identify them accurately. So in the first abstract, this is a novel genetic architecture for all-cause cirrhosis and hepatocellular carcinoma that was presented at one of the plenary sessions. The aims of this study were to really understand some of the, augment the current repertoire of genetic variants that contribute to cirrhosis development and HCC development. They wanted to evaluate the interplay between these genetic loci and underlying disease etiologies, which we won't have time to talk about during this talk. And a really exciting part of this is to talk about genetic risk scores and how they could be predictive of outcomes in these cohort populations. For this, they used a very large set of data sources from diverse backgrounds of patients. And you can see here that they found 8 loci that are shared between HCC and cirrhosis. Some of these were novel, some of these were known, but there were also 4 that were very specific to HCC as well. And in the genetic risk score, when they developed this, they could really risk stratify the population for development of HCC. That's what the axis here is on the y-axis, and the x-axis is over time. So you can see if you add these to the underlying liver etiology, you can increase your prediction of development of HCC. So they found 11 novel variants for cirrhosis and 4 for HCC. Interestingly, a lot of these were related to major histocompatibility complex class 2 alleles, suggesting some immune-mediated effects that may predict the risk of development of these. And they found this genetic risk score predicted the progression from cirrhosis, from chronic liver disease to cirrhosis and cirrhosis to HCC. And they found additional interactions with metabolic dysfunction with viral hepatitis and HCC risk as well. The next abstract is from Agrawal et al. This is really looking at patients with non-cirrhotic HCC, which is a significant population, but really understudied out there. This study shows that about a third of patients in the VA diagnosed with MASLD-related HCC had non-cirrhotic MASLD as an etiology. And so there's really sparse data characterizing the histological variants of non-cirrhotic HCC. So the group here want to really develop a kind of multimodal way of characterizing this using morphology-based molecular classifications, immunohistochemistry panels, and looking at somatic gene mutation profiles. So again, this is a rare population that's not been studied very well. This was from a group in India that included 40 patients for this analysis, the majority of which had MASLD as the underlying etiology. And again, they performed this multimodal evaluation. These are the top line results. And I think what the take home is here is that there's a lot of heterogeneity, but there can be some in the histotypes that we see, but there can be some, you can trace it back to some of the etiology and potential pathways that lead to HCC in these populations. And if you look at the genetics, I think the big message here is that TERT polymorphisms are a big driver of HCC, and it's true in this non-cirrhotic population as well. And so the conclusions here, again, where there is high heterogeneity, the TERT germline mutation is important. Microtrabecular are predominant in MASLD etiology and this beta-catenin pathway signaling and male predominance, suggesting a pathway for these patients to get this. And then this compact histotype, again, is well-differentiated mutation, beta-catenin signaling, and about more prominent in female. So these are different histotypes. I think it's a small study, but important. This is a really difficult subtype of HCC to study. The next study will really talk about risk prediction in HCC in patients who've been cured of hepatitis C from Kanzaki. So there's it's called the serum biomarker score to evaluate HCC risk in HCV-cured patients with cirrhosis. So we do know that patients who've been cured of hepatitis C have a persistent risk of HCC. This was nicely shown by George Awanu at University of Washington in the study from the VA, showing that there's a persistent risk even after SVR in patients with cirrhosis. And so what the study team here was really trying to do is trying to develop a risk stratification pathway for these patients. And so what they did was they looked at tissue, derived a signature, and then they developed that, based upon that, they developed a serum-based signature for HCC risk, and then they validated it. I'm going to show some of the validation data. This is the tissue signature validation, and you can see here that they've developed a really nice curve separation, and actually no HCCs developed in the low risk group. Again, small validation cohort, but powerful validation from the tissue signature. Now, when they looked at this in the serum, when they developed a secretome-based signature, they were, again, able to differentiate, not quite as well as you would expect from the tissue, but you can see here that the factors that are involved, including some of the demographic factors of the patients. So this hepatic transcriptome-based PLS HCV cure was developed and validated, both in tissue and serum. And again, this reflects some molecular dysregulations associated with necroptosis and TNF-alpha activation. The serum proteome-based PLSEC HCV cure was developed and validated. To what extent this can be used and implemented in clinical practice remains to be seen, but interesting data to see how we can risk stratify this high-risk group. The last risk prediction abstract is from the Bino John at the Miami VA, which looked at liver stiffness as an integration into risk stratification in MASLD. And so they really wanted to look at the patients who've gotten liver stiffness measurements within the VA to establish cutoffs or trying to figure out subgroups within the MASLD population that we can potentially identify as needing surveillance. So what they did was they used a large VA cohort, over 30,000 patients that had transient elastography and MASLD, and they were able to identify some subgroups that meet the threshold for potentially, being cost effectiveness for surveillance. Again, males, LSM between 10 and 14.9, excuse me, and those with diabetes. So every 5 kilopascal increase in LSM increased HCC risk by 19%. And again, HCC surveillance may be cost effective in males and diabetes. And so we can maybe integrate this in a way to personalize HCC surveillance and AASLD. This is, again, a VA population. It'd be nice to see a more mixed population, women, et cetera. But I think this is a really nice proof of concept. I think the next topic we'll cover is surveillance for HCC. The first abstract is from Dr. Kim at University of Washington, and talking about the optimal AFP threshold for HCC screening should be lower than 20 for cured Hep C, ALD, and MASLD. We do know that serum AFP is a part of HCC screening recommendations currently, integrated with ultrasound. Per the guidelines, AFP greater than 20 is considered a positive screening test, but we do know that AFP varies by liver disease etiology. In this study, they looked at 28,000 veterans, another large VA study, 1,000 cases, 27,000 controls, and they evaluated test sensitivity across etiologies of disease. And you can see here that Activate Hep C that has the highest sensitivity for AFP, but at different thresholds, you actually don't lose that much in other etiologies of disease if you drop the threshold further from 20 to 10, and you may increase sensitivity a bit. Specificity is the other concern when you think about shifting your cutoffs, and actually specificity is relatively well preserved in these patients who have cured Hep C, ALD, and MASLD. So AFP performs better as an HCC screening test in active HCV, which we knew, but in patients with cured Hep C, ALD, and MASLD, lowering that threshold from 20 to 10 would increase test sensitivity by about 10%, while maintaining that high specificity that you would want with a screening test. So I think this is a really nice study to show this, and I think a really large-scale way of evaluating AFP. The next study is about a novel way of testing or doing surveillance and using this multi-omics biomarker signature derived from circulating hepatocyte extracellular vesicles. Extracellular vesicles are an interesting way of performing surveillance. They're, again, circulating, and they are excreted by cancers, and they've been utilized in HCC and other cancers, but this is one of the first commercial companies to really come up with a way to look at this in a study. So these EV biomarkers, they have protein biomarkers off the EVs, mRNA biomarkers off the EVs, and then some soluble proteins, including AFP and DCP, and they were able to categorize Hep C or HCC positive and HCC negative based upon this. They presented nicely some of the derivation work that led to the study, but in this abstract, they really focused on the testing set, which is in cohort 2 here, which included 16 HCC cases and 17 patients with cirrhosis. And you can see here the AUC was very nice, 0.9, very small case control, kind of a phase 1 study, but a 0.9 is great, and it compared favorably to GAD, which is DCP, AFP, gender, and age in this small study. If you look across stages of HCC, it performed very well in early stage disease with high sensitivity and specificity, which is what you want to see in these early studies. This approach needs to be validated in larger phase 2 and phase 3 studies, which I know the company is planning, but this is a really interesting approach for surveillance. The next one, we'll talk about prognosis from David Goldberg, who's in a category of his own, as many of you know, here talking about prognosis of patients with HCC. This group was interested in developing an objective way to predict prognosis in patients with intermediate and early stage HCC, to really think about how you can talk about overall survival benefit of treatments, including liver transplantation. And existing ways that we do this right now are based on subjective criteria, including ECOG status. So they wanted to get rid of those subjective criteria. This is, again, another large VA study, retrospective cirrhosis cohort from the vocal group in patients diagnosed with HCC. They censored the patients at the time of transplant, but one of the good or bad things about the VA is that there wasn't a lot of censoring because there isn't a lot of liver transplantation. You can see the variables that were included here in the study, and these are the top line results, which showed that the AUC compared favorably to BCLC and ALB score alone. They did combine BCLC and ALB together, and it was very similar. Interestingly, 30% of the patients in this cohort were untreated, which I think their group is going to look at a little bit better or a little bit more. But again, this was more predictive in that group specifically, which is relatively understudied. Again, this presents an objective score to predict survival from diagnosis in a diverse cohort with early and intermediate stage HCC. Good performance, not outstanding, but pretty good, and I think it's difficult to predict mortality in this group of patients. But again, these are potential applications, thinking about expected survival, guide decision-making, and then combine these with post-transplant survival models to really think about what the survival benefit of transplantation is. The last two studies we'll talk about are HCC treatment. The first is from Ms. Bansal, who's a medical student and a second-generation AASLDer here. So this is really looking at patients with Child-Pugh B liver disease and receiving first-line immunotherapy, which is the STRIDE regimen. Durvalumab plus tremelimumab. So tremelimumab in this regimen is given out at the outset as a CTLA-4 inhibitor. The STRIDE regimen is an approved first-line option in patients with unresectable HCC. However, all the data in the registration trial were patients with Child-Pugh A. Child-Pugh B patients, we see a lot in clinical practice. It's a relatively understudied population of patients. And so the group here really wanted to describe the utilization and outcomes in STRIDE in unresectable HCC and Child-Pugh B cirrhosis. They included patients from the VA. It was a small number of patients, but 30 patients is, this is the first data in an IO plus IO therapeutic combination in Child-Pugh B patients that have been, presented in a multi-center fashion. You can see most of the patients had a B7 disease, some with B8, and a small number with B9, and the majority had non-viral etiology of their HCC, which is also important because there has been some controversy about the utility of immunotherapeutics in non-viral etiologies of hepatocellular carcinoma. We look at the survival. They compared it to the patients who had Child-Pugh A that received this. The Child-Pugh A patients, the median survival was not achieved by the time of this, but we do see that the median OS was 6.2 months in the Child-Pugh B patients. So not great, but certainly there are some patients that respond and have a prolonged survival. The other important part is how well was this tolerated? Grade 2 to 3 treatment-related adverse events happened in about 32% of patients. So actually pretty well when you look at the clinical trial data. And when they compared it to Child-Pugh A patients, the rate of immune-related adverse events was very similar. So, is this safe to give? Yes. Is it efficacious? I think it remains to be seen. You can see the numbers are pretty small as we get further out here. Greater than 25% of patients in the VA received STRIDE that were Child-Pugh B, and 2/3 received prior LRT. Most of these patients had microvascular invasion, or macrovascular invasion, and extrahepatic metastases. Again, 76% were BCLCC. They got about 2.5 cycles through was the median time, and again, the median OS was 6.2 months. I did not show this data, but there was some deterioration in liver function, but it did not happen until later. And so some of this is probably just due to progression of their liver disease or their cancer rather than a direct effect of the treatment. The last study was presented yesterday as a late breaker, as an encore from Josep Llovet from Mount Sinai. This is a LEAP-012 phase 3 study of lenvatinib plus pembrolizumab plus TACE for intermediate stage HCC. I think there's a lot of interest in combining therapies in earlier stage disease. We heard from GI ASCO last year of the EMERALD-1 results with the primary endpoint being PFS. This is another combination therapeutic regimen to look at lenvatinib plus pembrolizumab plus TACE in the treatment of intermediate stage HCC. It's a global study, randomized patients that were TACE eligible, 1-to-1 between lenvatinib plus pembrolizumab plus TACE versus placebo plus TACE. There were some various stratification factors that you can see listed here. And the primary endpoints were PFS and OS. And again, this is the first cut of the analysis. And so some of these are immature endpoints at this point. There are 480 patients assigned randomly, and you can see the flow charts here from the consort. The median time from randomization at this time of analysis was 25.6 months, so reasonable follow-up in this population. If you look at the primary endpoint of PFS, The study met its primary endpoint so far with a hazard ratio of 0.66, and you can see the confidence interval there. The median PFS in the patients who received the combo was 14.6 months versus 10 months who received dual placebo plus TACE. You can look at here, the objective response rates were also about 13% higher in patients who received the combination. Interestingly, the disease control, or excuse me, the duration of response were very similar here. So once you did get a response, the duration of response was similar, but the absolute number of patients who had a response was higher in the combination. The disease control rate, again, you can see that is higher here. AEs are always concerned when we start adding therapies on here, and the treatment-related AEs were a significant issue here, I think, in the combination, including those grade 5s, which we never like to see when we have patients on multiple therapies. And so I think as the data matures, we'll have to understand these patients this more. The serious AE event, again, was 2 to 3 times higher in the patients who received the combination. So in conclusion, this LEAP-0012 study showed a clinically meaningful and statistically significant improvement in the PFS. I think the side effects were manageable. However, I do have concerns about the serious and the grade 5 side effects. So I think we'll have to see how this evolves. Not certainly ready for prime time in clinical practice yet, but I think we're going to see more and more studies like this in this space. So with that, I thank everybody. I thank the organizers for having us present, and all the real presenters out there sharing their slides with me to facilitate this. So thank you all.
Thank you. Next, I'd like to invite Dr. Aparna Goel, who is an associate professor of medicine at Stanford University in the Division of Gastroenterology and Hepatology, and she will provide today's cholestatic liver disease debrief.
All right. Thank you to the organizers for inviting me to give the cholestasis debrief this year. It is a very exciting time for cholestatic liver disease, and it's an honor to be here. The journey for cholestasis, this meeting was very broad and comprehensive, so I will try to do my best to highlight the key abstracts. I want to thank all of the presenters, even if your work wasn't highlighted today in my meeting. And I think some of the satellite symposium, you saw the very long lines for lunch, so I think it was a very interesting programming. So I also want to thank all the folks that shared their slides with me in preparation for the debrief. This is really quickly what I'll try to cover in the 20 minutes that we have. We'll go ahead and get started with primary biliary cholangitis. So as you all know, PBC is a chronic autoimmune liver disease that's characterized by the destruction small and medium-sized intrahepatic bile ducts, predominantly affecting middle-aged women. And this year, we've seen the accelerated approval of 2 novel therapies for second-line therapy for PBC, elafibranor and seladelpar in the US. But before we understand how we're going to incorporate these novel therapies into our practice, we have to really understand how we're currently practicing in managing PBC. And this was examined by Dr. Ronca in the Fibrate-OCA study, a global snapshot of PBC around the globe. So in this study, this was a retrospective multi-center study that involved 40 centers across Europe, Japan, South America, and North America. PBC patients who began second-line therapy treatment with fibrates or obeticholic acid were included, and clinical biochemical treatment data at diagnosis and at the start of second-line therapy was captured in addition to adverse events and treatment changes. There were a total of 1,785 patients included in this study, 917 initiated on obeticholic acid and 868 on fibrates, 2/3 of which was bezafibrate, and nearly half of the patients that started on bezafibrate were from centers that had restricted access to obeticholic acid. 17 patients were started on combination therapy. And I think this waterfall plot really beautifully demonstrates the journey of our PBC patients that are started on second-line therapy, and something that we have to really keep in mind as we start to incorporate the new therapies that have been approved, the new PPARs that have been approved. So during the course of treatment, 10% of patients that were on obeticholic acid had to add a fibrate, and 2.5% of patients that were on a fibrate added obeticholic acid. And I think notably, the discontinuation rates for both of these medications is actually quite high, 33% in the obeticholic acid group and 20% in the fibrate group for various reasons. So this really highlights that the management of PBC requires dynamic adjustments to second-line treatments, and that there are high discontinuation rates that underscore the need for us to understand or create better allocation tools for which patients will respond best to which therapies. Now, second, another big area of discussion has been what our treatment goals for PBC should be. Historically, we've used a criteria of an alkaline phosphatase cutoff of 1.5 to 1.67 times upper limit of normal and a normal bilirubin to define adequate treatment response. But the question is whether or not normalization of alkaline phosphatase and normal bilirubin should be the actual threshold. This was examined by Dr. Reddy using the target PBC cohort, which is a longitudinal cohort registry of PBC patients across multiple sites in the United States. And patients were included if they had a recorded start date for UDCA, they had no second-line therapy at any point, and an adequate response to UDCA was defined by the PARIS-2 criteria, and patients had no prior decompensating events. So in this study, patients were stratified into two groups. Those that were defined as having a deep response, defined as either normalization of alkaline phosphatase or total bilirubin less than 0.67 times upper limit of normal, and those with an adequate response defined by the PARIS-2 criteria. And the primary outcome was complication-free survival. As you can see here, there were 232 patients that were included in the study. 82% of patients that had a deep alkaline phosphatase response also had a total bilirubin that was less than 0.6 times upper limit of normal, and about 50% of the cohort had advanced fibrosis. There was a significant decrease in the hazard ratio of developing serious complications in both the deep alkaline phosphatase and the deep bilirubin response groups. And it really appeared, as you can see with the Kaplan-Meier curves, that this improvement in survival or complicating events was really driven by the folks that had advanced fibrosis. So the authors conclude that patients with advanced fibrosis in particular are likely to benefit from more aggressive therapy to achieve normalization of alkaline phosphatase and a deep bilirubin response. So something to keep in mind as we see patients with PBC and really understanding risk stratification early in the course of their therapy and how to individualize treatment response as we have novel therapies. But is there a benefit to treatment if the alkaline phosphatase target cannot be achieved? And I think this is an important question. Dr. Codley presented data to answer this in the abstract, looking at obeticholic acid inflammatory markers from the POISE study. And reminder that this was a study with obeticholic acid that included 216 patients. There were 143 patients that were treated with obeticholic acid in either the titration arm or the 10 milligram arm. There were a total of 77 incomplete responders to obeticholic acid in the POISE study. And the specific biomarkers that they used included FGF19, CRP, IgM, CK18, FIB4, and APRI scores. And what they found was a difference in median percentage compared to the baseline in both groups that received obeticholic acid, either the titration group or the 10 milligram group. And there was also a trend toward improved fibrosis scores using APRI. So perhaps in patients that can't achieve a composite endpoint for alkaline phosphatase, there may still be benefit to obeticholic acid. Now, with a newly approved PPARs, is there a benefit or is it safe to use PPARs in PBC patients with cirrhosis? Dr. Villamil reported the efficacy and safety of seladelpar in patients with PBC and compensated cirrhosis from the phase 3 response trial that's been previously published. And just a reminder that the response was a phase 3 study with PBC patients that had an inadequate response to UDCA or that were intolerant to UDCA, and patients are randomized in a 2-to-1 fashion to receive either seladelpar 10 milligrams or placebo. There were 27 patients with cirrhosis that were included in this initial phase 3 study, 18 that received seladelpar and 9 that received placebo. So this study is small, but it does give us some insights into the safety of this drug in patients with cirrhosis. In terms of response, in general, patients that had cirrhosis had a similar reduction in alkaline phosphatase that was sustained through the study, and the percentage decline was actually quite similar to that seen in patients that did not have cirrhosis. And bilirubin levels remained relatively stable throughout the study in addition to INR and MELD scores. The safety profile in patients with cirrhosis was very similar to that seen in without cirrhosis. And importantly, there were no patients that had cirrhosis that discontinued seladelpar due to AEs. So the authors conclude that seladelpar decreased cholestatic and liver injury markers similarly to that seen in patients without cirrhosis, and it appeared safe and well tolerated in patients that had compensated cirrhosis. Is the biochemical response that we see with PPARs, there's been data presented up to 52 weeks, that was previously published, but is the response durable? And this is the last one that we'll review in PBC. So Dr. Cowley answers this in the presentation, looking at the long-term efficacy of elafibranor from the ELATIVE trial. And remember, similarly to the prior study design for response, ELATIVE was a randomized study looking at patients with PBC that had an intolerance to or inadequate response to UDCA, and were randomized 2-to-1 to receive elafibranor, which is a PPAR alpha delta agonist. So there were 138 patients that were entered into the open label extension of this trial, and 93 of which were continued from elafibranor from the double-blind phase, and 45 that crossed over from the placebo arm. And those that continued elafibranor, shown on the left there, you can see had an ongoing sustained improvement in their, or ongoing sustained reduction in their alkaline phosphatase. And the patients that crossed over from the placebo arm into elafibranor at week 52 had similar declines in their alkaline phosphatase that was seen in the double-blind placebo-controlled study. So let's shift gears to primary sclerosing cholangitis, a disease, a fibro-obliterative disease of the larger bile ducts that predominantly affects middle-aged men that usually have concomitant inflammatory bowel disease. An important area in PSC research is understanding other biomarkers to use and exploring and validating surrogate endpoints because we know that the alkaline phosphatase has suboptimal performance. So I'd like to highlight 2 studies on this topic. The first is an analysis from the phase 3 premise study that looked at associations between biomarkers and the MRI-derived ANALI scores. This was presented by Dr. Troner. So the ANALI score has been proposed to potentially predict disease progression in PSC. In this study, they evaluated baseline associations between the ANALI score and other non-invasive biomarkers in patients with PBC, and they also evaluated the ANALI score for the prediction of PSC-related events. And a reminder that the ANALI score is a score that's generated by looking at liver dysmorphy, intrahepatic biliary dilation, and portal hypertension from MRI findings. And it's calculated with these findings, and a score that's greater than 2 suggests higher risk disease. The phase 3 premise study included 419 adult patients with PSC, some that were in the placebo arm and some that received silofexor. And at baseline, you can see that the ANALI score was significantly associated with ELF score and liver stiffness measurements, and a higher ANALI score was associated with cholangitis events during the study. An ANALI score greater than 2 at baseline was associated with increased ELF score and increased liver stiffness measurements. And these associations, when they looked at the individual components of ANALI, these associations were really driven by dysmorphy and portal hypertension and not by the presence of intrahepatic biliary dilation. So the authors conclude there was also an association with ANALI score and the level of liver enzyme measurements as well. So the authors conclude that ANALI score, or the components of it, could become possible non-invasive biomarkers for selection into really trying to consolidate the phenotype of PSC patients that we select into our PSC trials, or potentially looking for outcomes in PSC trials as well. Another study evaluated transient elastography as a prognostic indicator in PSC. This was presented by Dr. Leung. This was a single-center study of real-world use of liver stiffness measurements, looking at as a dynamic prognostic indicator over a 10-year period. There were 403 patients with PSC, and there were 84 composite events during this study period with over 1,300 liver stiffness measurements. And the authors found that for every log increase in liver stiffness measurement, that there was almost a 4.5-fold increase in the risk of decompensation, transplant, or death. And when they adjusted this analysis for female sex, age at first liver stiffness measurement, and alkaline phosphatase and bilirubin levels, the hazard ratio remained quite elevated still at 3.25. And there's a nice example that I appreciated that was presented in the poster that really that looks at one single patient, same patient, age, what their alkaline phosphatase levels are, what their bilirubin levels are, same age, and really looks at how their risk of these complications changes based on their liver stiffness measurements. So this study validates findings from prior studies looking at liver stiffness measurements as a prognostic tool for PSC, and it could be incorporated into trial design as well. Now, 2 novel therapies that I'd like to point out for PSC, which is really exciting. We saw the novel therapies for PBC. There is a lot of work being done in the PSC space as well. The first is bexotegrast, which is an oral inhibitor of alpha V beta 6 and alpha V beta 1 integrins, and looking at markers of cholestasis and fibrosis. This was presented by Dr. Cowley. So the dual selective inhibitor of alpha V of these integrins, we think is a main mechanistic target in blunting the TGF-beta signaling, which we know is activated by the alpha V integrins and thought to play a key driver of fibrosis in PSC. So in the Integris study, patients with PSC and suspected fibrosis were randomized to receive either Bexo or placebo. And this abstract focuses on the final 24-week analysis of the safety and tolerability of Bexo 320 milligrams versus placebo, and looks at some exploratory endpoints as well. So there were a total of 27 patients in the Bexo group and 9 in the placebo arm, so still a small study. 2/3 of patients were on UDCA, and nearly half had IBD. The degree of fibrosis by ELF or transient elastography was similar in the two groups. And importantly, treatment emergent adverse events were similar between the Bexo and the placebo-treated groups. And there's a few interesting findings here. One was that Bexo did lower alkaline phosphatase values from baseline to week 24, compared to a slight increase that was seen in the placebo group. There was also a numerical difference in cholangitis and pruritus events observed between the two groups. And the itch NRS score was stable from baseline to week 24 in the Bexo group compared to an increase that was noted in the placebo group. And in terms of fibrosis markers, we saw stabilization of ELF scores through week 24. And in those that had a baseline ELF score of greater than 9.8, maybe a slight improvement. There was also similar stabilization noted in the transient elastography scores. And then lastly, there was some MRI tools that were evaluated, or MRI parameters that were evaluated. One was the overall whole liver enhancement of, in both the Bexo and the placebo groups, and there was improvement in the relative enhancement, and also the time to arrival of contrast into the common bile duct, which was improved in the Bexo group. So what we see is that this is a well-tolerated drug that's shown improvement in alkaline phosphatase and stabilization of symptoms associated with cholestasis, and lending support to longer, larger studies using this drug. The other late breaker was studying a novel therapy looking at CM101 in PSC. This was the phase 2 SPRING study. This was presented by Dr. Bolus. So CM101 is a humanized monoclonal antibody targeting CCL24, which has been implicated in inflammation and fibrosis. This was a phase 2 randomized placebo-controlled study of CM101 in patients in non-cirrhotic PSC. And there were 76 patients that were randomized to either receive CM101 or placebo, and the doses of CM101 varied between the randomization arms. Treatment emergent adverse events were mainly mild and distributed similarly across the cohorts, and there were no serious treatment emergent adverse events related to the study drug. So there was a consistent pattern, as you can see here, of a decline in liver biochemistries that was seen in the 20 milligram per kilogram treated group. And those were included patients that had advanced fibrosis. And interestingly, you can see that the effect on AST, ALT, and GGT was actually a little greater than the effect on alkaline phosphatase. There was also significant improvement in liver stiffness measurements in patients with moderate to advanced fibrosis treated with CM101, and ELF scores were improved in patients with moderate to advanced fibrosis that received the 20 milligram per kilogram group. And lastly, pruritus scores were lower in the CM101 treated group. So the authors conclude that this is well demonstrated, and that this is well tolerated, and that it demonstrated a dose-dependent anti-inflammatory and anti-fibrotic effect supporting further studies. Now, I want to spend some time just talking about, we've talked about disease control for PBC and novel therapeutics for PSC, but I think another important area is looking at the patient as a whole and talking about the management of symptoms, namely cholestatic pruritus and other health-related quality of life impacts. So this again should be considered part of our treatment goals when we're looking at cholestatic liver disease. And we know that despite the existence of guidelines and guideline recommended therapies, that the use of medications and guidelines is actually quite poor. But how big is the unmet need for cholestatic pruritus? So this was answered by Dr. Hirschfeld in his paper that was presented earlier, looking at the phase 3 GLISTEN trial and looking at the baseline characteristics of patients in the phase 3 trial. So GLISTEN, which stands for the Global Linerixibat Itch Study of Efficacy and Safety in PBC, is a phase 3 study of linerixibat, which is an IBAT inhibitor, so an ileal bile acid transport inhibitor, and compared to placebo in patients with PBC that have moderate to severe pruritus. So there are 238 patients that are enrolled in this phase 3 study. What we know is that 25% of patients have normal alkaline phosphatase. So the presence of liver biochemistry abnormalities does not necessarily predict whether or not a patient will have moderate to severe pruritus. And we also saw that 50% of patients were not receiving any therapy for their pruritus at the time of enrollment, and only 9% were receiving bile acid binding resins. This is despite its guidance as first-line therapies. So these findings really underscore the unmet need for approved treatment for cholestatic pruritus in patients with PBC, but they also highlight the importance of us as providers of really having discussions with our patients and assessing the severity of their cholestatic pruritus. There were several studies that looked at the impact of itch on quality of life. This was presented by Dr. Levy in the Itch-E study. And one of the things that the severity of itch was correlated with several patient-reported questionnaires in 21 patients that were recruited from PBC advocacy groups. And what we saw was that patients that had moderate to severe itch had worse fatigue, cognitive, and social indices, indicating poorer quality of life, again highlighting the importance of understanding pruritus and really treating these patients effectively. Now, how do the newly approved PPARs impact pruritus? So Dr. Kremer reports the impact of seladelpar in terms of patients that had moderate to severe pruritus in the RESPONSE trial. And in patients that had moderate to severe pruritus, there were several categorizations in terms of improvements with PBC-40 itch domains and quality of life as well, in 72 patients in this study. So these were 72 patients that had moderate to severe pruritus to begin with that were enrolled in the RESPONSE trial. 49 were enrolled in the seladelpar group and 23 in the placebo group. And we saw that seladelpar reduced the mean itch intensity, bringing it down from moderate levels down to mild levels using the NRS score. And in those that had severe pruritus, improvements in itch were associated with improved fatigue and improved sleep. And I think perhaps very interestingly in this study, we saw that patients that did not have itch at baseline of study did not develop itch at month 12 if they were enrolled in the seladelpar arm, compared with about a quarter that were in the placebo arm that did develop itch after. So seladelpar reduced severity of itch to mild levels, and it did cause resolution in nearly 20%, and it may potentiate the development of itch in those that do not have it. And really quickly, looking at IBAT inhibitors in terms of efficacy for the treatment of pruritus and PBC, Dr. Cowley presented the data for volixibat for cholestatic pruritus. And these were patients that were studied with either volixibat 20 milligrams, 80 milligrams, or the placebo arm. There were 10 patients in each arm that had an average itch row score of 6 to 7. We saw significant reductions in cholestatic pruritus using the itch row scores in both the 80 and the 20 milligram arms. And these findings will support, hopefully, future studies looking at the 20 milligram BID group for patients that have pruritus. And then I will skip this because I'm running out of time, and I want to quickly bring up one other big part of our autoimmune hepatitis. I don't think this will be covered in any other piece here. So I'd like to highlight just one key abstract in autoimmune hepatitis. And this is obviously one that didn't have a ton of therapeutic discovery recently, and we've been using the similar drugs for quite some time. This study was presented by Dr. Welch looking at anti-TNF therapy as a mechanistic target for autoimmune hepatitis. And there's two key components to this study. The first was looking at it as a mechanistic proof that's targeting TNF, and the second was looking at a phase 2 sort of proof of concept study for corticosteroid-free induction. So first, biopsies of patients with autoimmune hepatitis were analyzed by mRNA-seq and correlated with transaminases and histologic features. And this showed that transcription factors for TNF and JAK-STAT pathways were enriched in the AIH cohort, in addition to their respective cytokines correlating with disease activity. And the second part of the study was a trial in 12 patients with autoimmune hepatitis that were treated with corticosteroid-free induction therapy with infliximab for 24 weeks at varying doses. And the results of 8 of these patients are included and show that there was dramatic reductions in AST, ALT, and liver stiffness at 24 weeks. And all patients, all 8 patients, had AST levels and ALT levels that were less than 2 times upper limit of normal by week 24 of the study. So really maybe supporting potential targeting of TNF as an option for corticosteroid-free induction therapy in AIH, and more to come hopefully with that. So we covered a lot. It's a whirlwind tour for cholestatic liver disease and a glimpse of autoimmune hepatitis. I really think that there's a lot of discovery and happening in this area, and it truly is an exciting time point. So thank you so much for your attention.
Thank you very much, Dr. Gold. It really is a busy area, isn't it? So it's great to see. So we're going to move on now to alcohol-associated liver disease. And I'd like to welcome to the stage Juan Pablo Arab, who's an Associate Professor of Medicine at the Division of Gastroenterology, Hepatology and Nutrition at the Virginia Commonwealth University School of Medicine. So over to you, JP.
Thank you very much for the invitation and to the AASLD for the opportunity to present on ALD and Met-ALD. And I'm thrilled on the number of abstracts that are every year increasing in the ALD field. This was not the landscape 5 years ago. So I'm very happy for that. And I apologize with those that I was not able to include them on this debrief. We are going to briefly discuss on some preclinical abstracts. I know that Dr. Rothman already discussed some of them. Then we are going to talk about the clinical ALD, and then after on a very exciting abstract on the interaction between AUD and ALD. So the preclinical. We know about the importance of the gut-liver axis on ALD, especially, the interaction on the intestinal variant dysfunction and bacterial translocation going through the portal vein back to the liver. So in this interesting abstract, the authors found that the absence of intestinal mucin 2 promote global cell-associated antigen passages formation, and that way they saw that it prevents ethanol-induced steatohepatitis in mice. So again, I think here the concept is how we can target the gut, trying to protect the liver. So I think it's a very interesting preclinical finding. Another discussion, and we had many abstracts on this, was between, what is the role of neutrophils in ALD. I think it's a topic of controversy, because if you look at the circulating blood neutrophils, it's very different to what is happening in the liver, and even in the liver itself, it's different at different stage of ALD. So what they found here is that myeloperoxidase, it relates with outcomes, particularly with mortalities at 60 days. They didn't find any difference on other markers that were they trying to identify that were related to muscle metabolism. But if you look, in this rank, comparing patients with alcohol-associated hepatitis versus control, how neutrophil degranulation, and there's where myeloperoxidase have a role, stand up as a very important target. So I think we will need more investigation and more research in this field. GLP-1, I think everyone is very excited about GLP-1s, but this is not only MASLD. We know that there is some evidence that it may be a good opportunity in Met-ALD and ALD. And this is a mice model, so it's preclinical, but basically using semaglutide in a model of alcohol-associated hepatitis, they found that GLP-1 receptor agonists reduce injury, also the hepatic CYP2E1 activity, and increase antioxidant activity. So basically here the mechanism was, GLP-1 receptor agonists were reducing lipid peroxidation, the expression of some of the key inflammatories, also lipogenesis and oxidative stress genes, and also GLP-1 receptor agonists significantly downregulated CYP2E1. So I think more research is needed in this field. We know that also it's not only affecting the liver, it's crossing the barrier to the brain, so there is some reduction on behavior of alcohol consumption. But again, I think this is some molecular evidence that it makes sense to use semaglutide in this population too. We know that mitochondrial dysfunction is a very predominant feature in ALD, actually in alcohol-associated hepatitis. Some of the histological score that we have is looking at megamitochondria, but how this happened and the mechanism of this mitochondrial dysfunction is not very well known. So in this abstract, the author found that PDK4 has a role in promoting mitochondrial dysfunction in ALD, and basically by regulating the ER mitochondria interaction. So I think it poses, number one, the role of mitochondrial dysfunction in ALD, and number two, a potential mechanism that we could use to target mitochondrial dysfunction in ALD. This is a very interesting study using the UK Biobank, and I think this is a question that most of us has been trying to solve, like, how we can differentiate MASLD from MET-ALD, and what are the different profiles. So you can see here how in a very large cohort of 40,000 patients, and with the steatosis defined by MRI PDFF, being 11,000 with SLD, they did profiling, and they found that the top unregulated metabolites in MET-ALD were acetoacetate, the 3-hydroxybutyrate, and then HDL-associated lipids. And this is interesting because when you look, like the top 30, many of them were related to HDL. So whether this is just a consequence or it may be related to the pathways of injury and the mechanistic approach, I think that will need to be defined in the future, but I think it's an interesting finding. And also, another study was showing, the hepatic tracheostome, and usually we try to identify what is happening. But even the diversity of changes, so the alpha diversity analysis, it also related with, outcomes in this population. So looking at the global changes and trying to understand what is happening in the general transcriptome landscape can be an opportunity to identify novel pathways that could be susceptible of treatment. Let's go to the clinical part. So in the clinical side, I think tons of discussion around MET-ALD. I think one of the questions is, whether the feature of metabolic dysfunction affect outcome in ALD. And here the author from Japan, they found that the body mass index was related to outcomes, as well as, bilirubin and other things like returning to drinking after discharge in patients with a decompensated ALD. So I think some evidence that there is interaction with metabolic factors. Another paper very interested from the MGH group, 4,000 patients with ALD. If you look how many of them has at least one feature of metabolic dysfunction, is 97%. So we need to be careful because we may be defining more patients in the MET-ALD side that are actually ALD. And more interestingly, they found that diabetes is probably the only risk factor that is independently associated with progression of fibrosis and cirrhosis, and all the other patients were more similar to the pure ALD. Another one on obesity, but now in alcohol-associated hepatitis, so this very particular phenotype. It has been questions whether, having or not features of metabolic dysfunction or obesity affect outcomes in alcohol-associated hepatitis. So they found that patients with obesity had more severe hepatitis, and also, as you can see here, they did multiple inflammatory cytokines and also more of these circulating cytokines. So they pose here this concept that I think is interesting and will need to be further defined. But if we have MET-ALD, do we have something like MET-AH? I don't know if the answer for that, but I think that's the question that is posed there, and we will need more data. How we are doing with alcohol use disorder and ALD in the US? Well, not great, and this is a very interesting paper showing what is happening in the older population, and you can see in green the global trend, and then in orange what's happening in the US, how alcohol use disorder is increasing. And as you can see, this was even before the pandemic, so we are kind of blaming the pandemic, but these trends were before. How ALD globally is decreasing, but in the US, and this older population is increasing, same with liver cancer. So I think we know that this is a burden, this is an issue, and we need to keep acting and doing things. How we can assess early return to drinking in patients with alcohol-associated hepatitis. So this happened, 20, 30% here, but even in patients with moderate ALHEP, and actually those were the ones that had the higher rate of return to drinking. Why? Probably because we are paying less attention and following less closely those patients. So a call for action there in the patient with the non-severe ALHEP. Pharmacological therapies for alcohol-associated hepatitis is a study from Juan Abraldes in Alberta, and you can see how after the STOPA trial, there in green, pentoxifylline prescription almost disappear, but the corticosteroids keep increasing, although the STOPA was negative. And I think it's because there are some indication after. There is some evidence that corticosteroid may be of benefit at 30 days. So I think there has been some changes in the prescription patterns according to the trials. This is an interesting abstract from Dr. Luis Antonio Diaz showing that ACLF in ALHEP, it may be something that we need to be looking. So if you calculate, the score for ACLF in this population, you can see how the grade 3 has a discordant mortality compared to the MELD score. So maybe patients with ACLF grade 3 will need prioritization or assessment for liver transplantation right away instead of trying other pharmacological treatment. AHEAD is a machine learning model by Dr. Ashwani Singhal trying to identify, I don't know why it's going alone, trying to identify diagnostic criteria. So this was based on biopsy, but is with non-invasive clinical features and laboratory tests, and you can see how they outperform the NAAA criteria. So the NAAA criteria, AUC, was almost 60%, and this is 70%. It's not perfect, somehow better, and it's also a non-invasive test. So again, GLP-1s, so what is the effect on the risk of adverse liver outcomes in patients with ALD, but with diabetes? So you can see how if you treat with GLP-1s, patient with diabetes but ALD, it reduced 40% of the adverse liver outcomes. So again, more evidence that GLP-1s may be an opportunity in this population. Then treatment access and access to liver transplantation for ALD. So this study from California showed that, predictors for listing were mainly male sex and private insurance. And of course, the limits for sobriety and other things were very center dependent. And the most common reason for listing patients were psychosocial. So again, I think we have some social determinants of health that we need to work on the ALD population, especially in the context of liver transplantation. Another interesting study, long-term outcomes in bariatric surgery in patients with met-ALD. So long story short, the primary outcome was incidence of decompensated cirrhosis. There was no difference, although some benefits on the weight loss. So again, be mindful that in the population with met-ALD, the weight loss may help with the outcome of weight loss and maybe cardiovascular outcomes, but from a liver perspective, it's probably not beneficial. This was a late breaker last night, so if you were here, a very interesting study trying to challenge the concept of using 4 weeks of 40 milligrams of corticosteroids versus using 40 milligrams for 1 week, 30 milligrams for 1 week, 20, and then 10, and then stop. So reducing the exposure to corticosteroids. So an open label, but with a decent size of 127 patients per arm. And basically, what they found was that although there is no difference In mortality, there was significant less infections. So you can see the significant difference in infection rates, and then mainly for enterobacteria. And of course, less prescription of antibiotics. So they showed that the rapid tapering is reducing severity of infection with similar MERS goal, but no impact on survival. Larsucosterol, so we are all very excited with seeing what happened with this drug. This is the initial design. If you look the global intention to treat, there was a trend there that was not significant. When they present only the US data, you can see that it was significant. So the question is why? And one of the answers may be the time to hospitalization to treatment. You can see how in the US it's only 5 days, but in Europe was 13 days. So again, I think we need to better understand what are our practice. It may be related to the requirement of liver biopsy, for example, and waiting for the results of the biopsies. So I think it's a very good food for thoughts. So this is saying, showed in the graph how the global versus the US data changed in the number of days before getting the treatment. Alcohol use disorder to finalize and in ALD. So this is a very nice study from Dr. Skladani from Slovakia, showing that how audit and PET correlates. It's in micromoles per liter, so I put there the conversion for the people from this side of the pond. So there is some correlation, but it's not perfect. So again, I think the main concept here is be careful of using PET as an standalone test, and that PET does not mean alcohol use disorder. PET means recent heavy alcohol consumption. So I think probably the conclusion here is we should use both, because only using PET is, or only using audit is a miss catching 25% of heavy drinkers. And then this is a study from Michigan using, a brief intervention that was, the doctor doing the transient elastography followed by brief intervention, how that affect outcomes. It does, and a lot. If you can see here how patient change in the WHO level of alcohol drink, they significantly reduce alcohol drink after this intervention. So I think patients trust the hepatologist when you have an objective measure like transient elastography. It's a very good opportunity to do brief intervention, and this may be impacting on outcomes. Varenicline is another, drug that it may be helpful in patients with ALD and met-ALD. So here they analyze craving, and you can see how it was significant in reducing episodes of craving. So I think in the met-ALD population, it was a good opportunity. We will need to see what happened after. Naltrexone, so this is for patients with met-ALD, and here the big concept was that naltrexone resulted in reduction in decompensated cirrhosis, even in patients with met-ALD, highlighting that, our capacity of identify patients with alcohol consumption, even in the met-ALD, is not great. So this patient still benefits from medications for AUD. Here, using Trinatex, which is a very big database, the authors showed that the use of naltrexone was associated with lower rate of AKI, SVP, and mortality in patients with decompensated cirrhosis and ascites. So it's not only safe, it's probably also effective. So again, a call to use this medication and treat these patients. Claire-Durkin here show us that, early pharmacological treatment of alcohol use disorder and psychiatric consultation were associated with improved overall survival in patients admitted with severe alcohol-associated hepatitis. You can see the Kaplan-Meier curve, how impactful with not just HR of 0.26. So there is always questioning, how effective are these interventions. Well, I think at least in this population, they are very effective. And then another concept on what is, the side effect of these drugs. So I think the sulfiram has significant side effects. So we should start discussing whether we should recommend not using the sulfiram in our population. But acamprosate and naltrexone in different formulation is very safe to use, being the main side effect, gastrointestinal and nausea. And then integrating hepatology and addiction medicine consultation is also another way. So this is the MGH experience showing that it reduced the likelihood of further hepatic decompensating events. And lastly, I think this is a very cool study using some virtual reality to engage on cognitive behavioral treatment for this population. And that is not that judgmental, and the patient can use it very easy. So I think this will be some creative, out-of-the-box ways to engage this population. And I would like to use the opportunity to invite you to participate in our ATC on Met-ALD. It's going to be February 27, March 1st in Las Vegas, Nevada. Thank you very much.
Thank you. Next, I'd like to invite to the podium Dr. Alina Allen. She is a hepatologist and the director of the Maslow Clinic at the Mayo Clinic in Rochester, Minnesota. She will present the Maslow Debrief.
Thank you very much for the invitation and for this opportunity. These are my disclosures. I'd like to start with a disclaimer. Maslow abstracts on the clinical realm comprised about 500 from the about 2,500 total abstracts in this meeting. So I had the great privilege to sift through all of them and choose what can be presented in 30 minutes. So there were massive scientific gains, and I apologize to those that will not be mentioned here for this reason, but I know there's impactful science out there which will not make it to these slides. I'd like to congratulate all researchers and also to thank you to the authors for sharing their slides for this presentation. So what are the major themes in 2024 Maslow research at AASLD? If you're looking for an elevator speech for the question, what was hot in Maslow in 2024? Here it is. It was a lot about omics, from genomics, metabolomics, proteomics, much needed data in pediatric Maslow, which I was very happy to see. Of course, data on drug therapies continue to dominate the space as we need therapies here. And there were two flavors of this: available drugs from completed trials and real-world experience, and also new MASH-specific drugs making their way to the finish line. And I'll leave this towards the end of the talk. So let's start with the area of genomics, and I'll start with resmetirom effects on NASH with liver fibrosis in patients with NASH genetic risk alleles. This is a study presented by Dr. Naga Chalasani. And I think it is important, now that we have resmetirom in our clinical toolbox, to know whether we need to be concerned about a patient's genetic makeup. This is a post hoc analysis of the Maestro-NASH trial, which by now you are all familiar with. This studied resmetirom 100 milligrams and 80 milligrams compared to placebo. The paper has been published in NEJM earlier and led to the approval of this drug. What this study did was testing the SNPs in PNPLA3, HSD17B13, and TM6SF2, as well as other genes, but I will focus on the ones mentioned here in red. These were genotyping patients consenting to DNA collection in this study and genetic testing for the response to resmetirom on serial liver biopsies, and MRI-PDFF was analyzed. The biopsy, the PDFF, and other biomarker responses were analyzed within each treatment arm, comparing wild type, heterozygote, and homozygote for each genetic risk alleles. The outcome of NASH resolution is what I'm showing here, and what you see are bars for proportion of responders to 80 milligrams and 100 milligram of resmetirom in orange bars and placebo in the blue bars. And these are stratified by genetic phenotype. So on the PNPLA3, you can see that the responses between those with no mutation, heterozygote, and homozygote mutations was essentially similar. The same story was seen in HSD and TM6SF2 genes. So there was no apparent impact in genetic risk markers on NASH resolution in resmetirom and the placebo population. For the fibrosis improvement outcome, the data is presented in the same way, proportion of responders here, and the message is similar. There's no apparent impact in genetic risk markers on fibrosis improvement in resmetirom and placebo. So to me, this was good news. I think we can be agnostic to genetic makeup for resmetirom treatment. What about tirzepatide? This is a study that I will present just a few big line summary features of, because you will see this presented by Dr. Arun Sanyal in the late breaker. But this study looked at the PNPLA3 variant, specifically in those treated with tirzepatide. And this is another post hoc analysis from the Synergy NASH phase 2 trial, which evaluated tirzepatide in 5, 10, and 15 milligram doses compared to placebo in adults with MASH and F2 and 3 fibrosis. Again, the paper was published in New England Journal earlier this year. In this specific study, we look at 131 participants who completed the study on treatment and had PNPLA3 genotyping. These are the numbers, 33 wild type, 65 heterozygote, and 33 homozygote for the G allele, which is the one with higher risk. The treatment groups were pooled on the tirzepatide doses and compared to placebo by genotype for MASH resolution, fibrosis improvement, changes in liver fat by MRI-PDFF, and changes in body weight. From the MASH resolution perspective, what you see here is a proportion of responders in placebo in gray and tirzepatide in blue. And this is the overall response, and this is the response by PNPLA3 strata. And as you can see, there was a signal for greater tirzepatide treatment effect in those with CC genotype versus those who had G allele. This was not seen in those in the outcome of fibrosis improvement, which seems to be similar across the strata. Also not seen in the outcome of more than 30% fat reduction by MRI-PDFF, and also not seen in the responses in body weight. So this study concludes that tirzepatide treatment was associated with improvements in MASH disease activity, fibrosis, liver fat, and body weight across all 3 PNPLA3 genotypes. However, the tirzepatide treatment effect for MASH resolution was greater in those with the CC genotype compared to the G carriers. Fibrosis improvement, reductions in liver fat and body weight were similar across. So these data suggest that the G risk allele may be related to MASH independently of its effect on liver fat. Again, there were 3 GG carriers. I think this is an important signal that, in my opinion, needs to be studied in larger cohorts, but it's very interesting. Staying in the realm of genome, this study presented by Dr. Naga Chalasani looked at associations between MASLD and biological age and its acceleration as assessed by methylation clock. I chose this study because of the recent emerging data correlating metabolic diseases, including MASLD, with senescence and aging and degeneration. In this study, they aim to examine the relationship between MASLD and biological age and its acceleration in a cohort of multi-ethnic adults, and to assess the interactions between the biological age and its acceleration and PNPLA3 risk allele on MASLD risk. It evaluated 885 participants in the multi-ethnic study of atherosclerosis, or MESA. The MASLD diagnosis was based on CT images of the liver, and there was also methylome analysis in the circulating DNA. There were 4 different methylation clocks that were assessed. They found significant relationship between biological age and biological age acceleration by one of those clocks named Hannum. And what I show here on the left is the biological age and MASLD associations, and on the right you can see the biological age acceleration and MASLD association. The difference between the models is listed here. Note that the impact or the association decreases the more elements are included in the model for adjustment. There was also an evidence of interaction of PNPLA3 risk allele with biological age, noted here on the left, and its acceleration by Hannum clock, noted on the right. So the study concludes that biological age and its acceleration by Hannum are significantly associated with MASLD. There is an interaction between PNPLA3 G allele and biological age and its acceleration for increasing the risk of MASLD. Whether this interaction promotes MASLD progression and increases the risk of liver-related events needs to be examined and further studied, but I think this is very interesting. Moving to proteomics, you saw a glance on the previous presentation. As we're starting to scratch the surface in MET-ALD diagnosis and distinction between that and MASLD, this is a study, again, looking at the UK Biobank presented by Dr. Niharika Jakar, in which over 9,000 patients had MASLD and almost 900 patients had MET-ALD. Note that MET-ALD here was defined based on self-reported questionnaires, so there's a caveat there. They assessed the protein levels for comparison across individuals utilizing normalized protein expression, or NPX. They saw signals that can potentially differentiate proteins among MASLD versus controls here on the left, and between MET-ALD and controls. Some were upregulated, some were downregulated. I'll leave details more for the paper, but I think the signal is that there could be distinct proteomic profiles that could differentiate MASLD from MET-ALD. Retinol binding protein 5 and oxytocin neurophysin 1 were found as potential MET-ALD markers in this study. Plasma proteomics, in addition, can identify high-risk patients. And I think this is important data to consider, with the caveat that it needs to be validated in MET-ALD defined by biomarkers rather than self-reported questionnaires. So definitely more to come, I think, in this space. We're going to metabolomics here in a very special population, that of children. This is an analysis presented by Helena Huneau, who's a PhD candidate in Dr. Miriam Voss's lab at Emory University, who had a fantastic presentation. And she used an unsupervised machine learning analysis of youth enrolled in NASH CRN. The objective was to identify MASLD metabotypes in children. And they used an unsupervised machine learning clustering analysis using clinical and laboratory markers that associate with MASLD pathophysiology. They also used high-resolution metabolomics analysis for the metabolome profiling in the serum. And the hypothesis was that there are distinct MASLD metabotypes driven by inflammation, insulin resistance, and lipid partitioning. What they found was 3 clinically distinct metabotypes in pediatric MASLD. The top listed here in green is what they called early mild, which is characteristic of children that tend to be younger, have a lower HOMA2-IR score, a lower triglyceride and LDL level. The second metabotype was the adipolipid SBP in children with a higher waist circumference, a higher level of VLDL, higher systolic blood pressure, and higher uric acid levels. A third type was the inflammatory fibrotic, with a higher signal for liver disease severity, as illustrated by higher ALT, AST, and on histology, a higher proportion of MASH and advanced fibrosis. How could this impact practice? One thing to mention before we move is that the higher proportion of Hispanic ethnicity in those with early mild and inflammatory fibrotic phenotypes. So how could this impact practice in the future if we were to consider this for clinical care? Those children with an early mild phenotype could be focusing more on prevention. Those who have an inflammatory fibrotic phenotype and thus are at higher risk of hepatic complications would have a care pathway that would focus on nutrition therapy to decrease inflammation, avoid sugars and fructose as well as alcohol when of age, follow up by hepatology, and also maybe focus on antifibrotic medications if these will be available. soon. For those with adipolipid SBP phenotype, the risk may be higher in extrahepatic complications in cardiovascular disease. Therefore, the care pathway there, in addition to the nutrition therapy for obesity and heart health, would focus on early CVD surveillance and maybe metabolic drugs such as metformin, GLP-1 agonists, and statins. So I found this interesting. I think further research is needed to validate these findings and to disentangle these pathways and amino acid, energy metabolism, and microbiome links to these. Moving on to GLP receptor agonists and MASLD, also in pediatrics. This was presented by Andrea Tao at the Children's Hospital of Philadelphia. And this is a real-world study which had the objective to determine the effect of GLP-1 receptor agonists on ALT reduction in pediatric MASLD. It was a single-center retrospective chart review of patients younger than 21 who were treated with GLP receptor agonists between 2018 and 2024 with a diagnosis of MASLD. Of course, the treatment indication was obesity or diabetes. The endpoint values were collected up to 13 months post-therapy. They found 111 children with a median age of 15, treated for approximately 13 months, and the results were that ALT improved by 23 units at 6 months and by 18 units post-therapy. And the results are seen here, and I'm highlighting ALT, which started around a value of 78 and ended up around 62. There were also significant improvements in AST, GGT, triglycerides, and hemoglobin A1C, but interestingly, no improvement observed in weight or BMI. Why? That remains to be determined, but I thought it was an interesting observation. There were even greater reductions in ALT in patients initiated on GLP-1 for an indication of type 2 diabetes, as you can see here. And among the GLP receptor agonists, semaglutide was the most common one, followed by liraglutide, dulaglutide, and exenatide. Side effects reported in 21 children, 5 had to discontinue the medications. So this is the largest dedicated pediatric study evaluating the effects of GLP-1 on MASLD to date. The treatment was associated with significant improvements in mean ALT, AST, GGT, triglycerides, and hemoglobin A1C. So by using ALT as a surrogate for histologic improvement of steatohepatitis, with its limitations, GLP-1 receptor agonists show promising results in treatment of pediatric NAFLD, may be most beneficial in those with type 2 diabetes. So further prospective studies are needed, and they are highly sought in the pediatric population. We're staying in the realm of GLP-1 in the real world, this time in the adult space. This is a study presented by Dr. Sydney Barrett. And this is a study of target NASH, a very large cohort of patients with MASLD and MASH of over 6,600 participants. Among these, you can see the breakdown of the over 4,000 patients who met the analysis criteria. You can see it here, and you can see the number of patients by disease severity, a very large proportion of patients with cirrhosis, I would say. Of these, almost 10% were GLP-1 receptor agonist users. Again, a lot of people with cirrhosis. The median duration was between 2.5 to 3.3 years, respectively, on these phenotypes. What they found was that disease progression, as defined by progression to decompensated cirrhosis, was in non-users of GLP-1 was 1.69 times the hazard of development compared to the GLP users. In the outcome of all-cause mortality, they found that non-users of GLP-1 had a 2.3 times higher risk of death compared to GLP-1 receptor agonist users. So I think important data to find from real world. I think there are caveats and healthy user bias and maybe other potential confounders, but I think important data to know. So we're going into resmetirom, back to resmetirom, but looking into a different angle here. And this was a study presented by Seth Bound looking at liver enzymes reduction from baseline over time in resmetirom treating patients in Maestro-NASH study. This was a poster, which I selected because I think it has impact specifically from the question of statin. We know that there's the label has this warning on a maximum dose of certain statins to be used in conjunction with resmetirom. So here they looked specifically at the 966 patients enrolled in Maestro-NASH, of whom almost half were on statins. And they assessed the direction of ALT in all patients, seen here the top graph, and then when split between patients on statin and patients not on statin, and specifically looking at ALT levels. What they found were that what was known from the label is that there are liver enzyme elevations in the first 4 weeks, specifically peaking at week 4, which occurred as a delta much larger in those on statins, but they were not associated with hepatotoxicity, and they're likely a reflection of lipid changes in the liver that occur adaptively. The statin exposure was within the range of normal variation, not different from placebo. There were no safety issue, and 98% of those doses were within those recommended in the label. But what I'd like to draw the attention more is beyond this delta increase in those on statin is the fact that patients on statin started at a lower ALT and AST, so that the peak really between these two groups is about the same in absolute value. So if you see this in your clinical practice, expect it and know that it does decrease with time, and it's not really a sign of hepatotoxicity irrespective of statin exposure. The efficacy on biopsy, PDFF, and lipid endpoints in patients on statin was equivalent to patients not on statins. Okay, we are moving to the exciting space of clinical trial data, and I will start with a study presented by Dr. Mazen Nureddin on efruxifermin, and which shows that significantly improved liver fibrosis at week 96 in the HARMONY study across subgroups, and improvements were associated with changes in biomarkers. So efruxifermin is a FGF21 analog administered subcutaneously weekly, which was studied in the HARMONY trial, and the data was presented at week 24. Current study extends these endpoints at week 96, so this is what we will focus on. And what you will see is a comparison between placebo and the different doses of efruxifermin, but also comparison between weeks 24 and weeks 96. So when we look at the outcome of fibrosis improvement by one stage, noted here on the left, you can see that 75% of patients on the higher dose of 50 milligrams achieved this outcome at week 96. So much deeper and more sustained response compared to what was seen at week 24. In the intention to treat analysis, which considers those who did not have a follow-up liver biopsy as non-responders, the differences were 19% in placebo and 49% in efruxifermin 50 milligrams. They used also digital pathology as a comparison to conventional histopathology, and as you can see here, the response rates are again very high, 81%, and what I found interesting was that this would have been caught actually at week 24 as well, so the delta here, or the time, wasn't as high as in conventional histology. There is evidence of disease reversal after 96 weeks on looking at other outcomes as well. Here you see a 2-stage fibrosis improvement, which was achieved by 36% of patients on the higher dose compared to 3% of patients at week 96. There were very good signals of response in biomarkers such as ELF or VCTE or combinations thereof, as you can see here. And they looked at disease response in all sorts of ways, from partial reversal to substantial reversal, this defined here, to near complete reversal, which means improvement to F0 or 1, and MASH resolution and liver fat content normalization. And this was achieved by 30% of patients in the high dose versus 0% in those on placebo at 96 weeks. So this is unprecedented antifibrotic activity observed in efruxifermin after 96 weeks. 3/4 of those receiving the 50 milligram dose experienced fibrosis improvement without MASH worsening, with early fibrosis response noted at week 24 and sustained and expanded through week 96. The consistent antifibrotic response was seen across high-risk groups such as stage 3 fibrosis, type 2 diabetes, PNPLA3 risk allele carriers. Conventional pathology corroborated by AI-based pathology, imaging, and circulating biomarkers all showed the same signal of improvement. And almost half of the patients on 50 milligrams of the medication were responders by all 3 measures of fibrosis: histology, ELF, or liver stiffness, which is very encouraging. I hope that as we move towards a biopsy-free space in clinical trials, this study will serve as really good data. Lastly, I didn't present this, but the acceptable safety and tolerability profile was also noteworthy, mostly with mild to moderate and transient GI events. So this year, late breaker session in MASLD is after this. So I will not be presenting details, so please stay tuned. It's going to be in the same room following this session immediately after. So I'll give you a flavor of what you are about to see, mostly with trial design and what was published for just preliminary results. So one of the studies will be presented by Dr. Mazen Noureddin, and it is once monthly efimosfermin alpha by Boston Pharmaceuticals in MASH with F2 and 3 fibrosis. These are results from 24-week randomized double placebo phase 2 trial. And recall that this is a long-acting FGF21 administered monthly. So I'm very excited that we can actually are at the stage to discuss therapy for MASH administered once a month. You can see here the study design, and again, you will hear more after this on the 24-week histologic response on this drug. And I will save, and I have saved the best for last. This is the study that was awaited this year. Semaglutide finished the line, the long line to success from the phase 3 ESSENCE trial in patients with MASH and moderate to advanced fibrosis. The 2.4 milligram dose met both primary endpoints of histological MASH resolution and fibrosis improvement. So stay tuned for this highly anticipated presentation given by Dr. Phil Newsome right after this. There are some other very noteworthy late breakers. The first one is the result of the 52-week phase 2B Voyage trial of VK2809 in patients with biopsy-confirmed NASH and fibrosis. This will be presented by Dr. Rohit Lumba. Another agent from Aligos, another THR-beta, similar to the first presentation, in a 12-week trial in non-cirrhotic MASH, phase 2A, also presented by Dr. Rohit Lumba right after this. And ervogastat, a DGAT2 inhibitor alone or in combination with clesacostat, an ACC inhibitor in adults with NASH, and this will be presented by Professor Quentin Anstey. So we had an amazing year for MASLD research in 2024. I was very excited to review all this and present this to you. If we were to summarize the take-home points we'll take with us as we get on our planes later today, what we saw this year will be the much-awaited phase 3 trial completed of semaglutide for MASH F2 and 3. Very noteworthy results in other phase 2 trials of FGF21 analogs, again administered weekly or even monthly, THR-beta analogs that I mentioned earlier, and DGAT2 inhibitors. We learned that genetic background does not impact response to resmetirom treatment, but the PNPLA3 G allele appears to decrease MASH resolution in tirzepatide treatment. We learned that biological age and its acceleration are associated with MASLD with a noted interaction with the PNPLA3 G allele. We saw early data suggesting signals of proteomic profile differentiating MASLD from MET-ALD. We saw distinct metabotypes in pediatric MASLD with potential impact in treatment pathways of the future, and I would love to see this in adults as well. And we also saw real-world data on the favorable impact of GLP-1 receptor agonists on MASLD activity and progression in both children and adults. With this, I thank you, and stay tuned for the late breakers.
So with that, I'd like to thank all of the presenters for the excellent and informative